Good morning, students. Today we will start with the next topic, that is regulation of gene expression. So, before starting the topic, we should know that what is the meaning of this sentence, regulation of gene expression. Regulation mainly means the type of control. So, here in this topic, we will learn about the control of the gene expression. We all know that genes, they are expressed, they are basically the proteins which are expressing themselves. So how these genes expressions, they are regulated at various level, we will study in under this topic. So what kind of regulation of gene it takes place in the different organisms? So what is the actual meaning of regulation? That is amount and time of formation of gene product as per the cell requirement. It means that some cells, they require a different kind of gene product and different amount at different timings. Okay, so different cells, they have their different requirements, so there is the need of regulation of the gene expression. So if we talk about the eukaryotes like us, humans, mammals, the gene is regulated at 4 levels. This is important question. Okay, so in eukaryotes, the regulation of gene expression, it takes place at 4 level. That is, first one, at it is the transcription level. Okay, transcription level, we all are familiar with the word transcription. Transcription is basically the process where the primary transcript is formed. Okay, mRNA is formed after the processing, and before processing, at the end of, basically at the end of transcription, hnRNA was formed. hnRNA was the heterogeneous RNA which is containing both exons and introns. So at that level, the first regulation is required. The second regulation is required during the processing level, when this hnRNA is converted into mRNA, that is after the processing. Okay, we have done 3 types of processes in the processing level: capping, tailing and splicing. So during that time, that again regulation is required. Next one is transport of mRNA from nucleus to the cytoplasm. Now the mRNA is transported from nucleus to cytoplasm for further synthesis of protein, because ribosomes, they are situated in the cytoplasm. Okay, so at this level, at this transfer level, again regulation is required. Finally, the fourth level is translation level, where in actual the protein is being synthesized. At this level, again regulation is required. But if we compare the regulation between eukaryotes and prokaryotes, eukaryotes, we are having the 4 levels where the gene regulation is required, but in prokaryotes only the transcription initiation. Okay, it is the predominant site for control of the gene expression. यानी यहाँ पर it is most important step. The transcriptional initiation process is that important process where the gene regulation is required. So in your syllabus, there is the regulation of gene expression that is undergone by the help of lac, that is lactose. So the model that is in your syllabus is lac operon model. Okay, so this lac operon model, before starting it, we should know some of the basic terms that are related with the lac operon model. Lac stand here for lactose. Lac here means lactose. So here we will discuss about the operon. So what is an operon? The concept of operon was first proposed by Jacob and Monod in 1961. It is quite important question for your NEET exam, that who had given the lac operon model firstly, who had proposed the lac operon model. So your answer is Jacob and Monod. They both are responsible for proposing the lac operon model. So what is an operon now? Okay, An operon, it is a unit of prokaryotic gene expression which includes coordinately regulated genes. Coordinately regulated genes means many of the genes, they are working together, okay, and control elements which are reorganized, which are recognized by the regulatory gene product. Operon, it is a specific functional unit, okay, which is controlling the gene expression, clear, which is controlling the and regulating the gene and its control, okay. So what are the different components of the operon? That is again important question. So now we will discuss about the components of operon. The first one is the structural gene. As the name is suggesting us, structural genes. So that is the fragment of the DNA which in actual transcribe the mRNA for the polypeptide synthesis. That is the part of the DNA which is in actual forming the protein chain, okay. Then comes the promoter. Again the name is suggesting us the meaning. Promoter means it is that part of DNA, that sequence, that fragment of DNA where RNA polymerase it binds and initiate the process of transcription of structural gene. As we know that structural genes, they are responsible for synthesizing the new protein, okay. So promoter is initiating the transcription of the structural gene so that they can easily convert into the proteins later on. Now come to the operator. The next component of operon is operator. It is again the sequence of DNA which is present adjacent to the promoter, just nearby to the promoter, and where the specific repressor protein it binds. Operator, it is the special kind of DNA sequence where the repressor protein comes and bind to, and then what happens, if the repressor will bind with the operator, the lac operon will get switched off. It will not work further. Now come to the regulator genes. The genes that codes for the repressor protein and that binds to the operator and suppress the activity, as a result which transcription will be switched off. I have already discussed with you in the upper point only, in the operator point, that operator, if it will bind with the repressor, it will switch off the model. It will switch off the process of transcription. No transcription, no translation will take place then. So regulator genes, they code for the repressor proteins. Okay, they will form the repressor protein which will further bind with the operator to switch off the model. Now just opposite of repressor is inducer. Inducer, as the name is suggesting us, it will induce, it will stimulate the process of transcription. So what is inducer? The substrate that prevents the repressor from binding with the operator. Inducer is that chemical which will bind with the repressor and make it inactive so that it is unable to bind with the operator. Now operator will remain free, and as a result what will happen, transcription it takes place, further translation will take place, and now we can say that in the presence of inducer the repressor cannot bind with operator and the model is called as switch on model. Now here one important thing that you have to remember is the regulation by lac operon, as it is controlled by repressor. As repressor is helping in switch on or switch off of the lac model, so that's why it is called as negative regulation, okay, because repressor is controlling the switch on and switch off of the lac operon model. So basically the lac operon model, it consists of three structural genes named as Z gene, Y gene, A gene. One operator gene which is represented by O, one promoter that is represented by P, regulatory gene, it is represented by small I. Okay, we have already discussed what is the function of regulator gene, to form the repressor protein. Clear? So lactose over here is the inducer which will switch on the lac operon model. Now there are two situations. First situation is when there is absence of inducer. Inducer is lactose. If lactose is not present in the medium, then what happen? Now I gene, that is the regulatory gene, it will form repressor mRNA which will further form the repressor protein. This repressor will bind with the operator and stop this operator, switch off this operator. Okay, so repressor bind to the operator region and prevent RNA polymerase from transcribing the operon. As here the operator, it will get switched off, then what will happen? The Z, Y and A genes which are coding for different kind of proteins, they will not be formed. So here there will be no transcription as well as no translation. Clear? That was the case of absence of inducer. Now if we talk about in the presence of inducer, inducer over here is, I am again repeating, lactose. Now lactose enter into the story. Now as the lactose it is present, what will happen? This lactose, it will bind with the repressor that was formed by regulatory gene. As this inducer it binds with the repressor, it will stop the repressor to bind with the operator gene. Now here the operator gene, it will remain functional, it will remain switched on, and as a result what happen? These structural genes Z, Y and A gene, they will undergo transcription as well as further they will undergo translation. Now the work of Z gene, as it is coding beta galactosidase enzyme, this beta galactosidase enzyme, it will help in the breakdown of lactose into galactose and glucose. What is the function of beta galactosidase enzyme? It will help in the breakdown of lactose into galactose and glucose. Now Y gene will also get transcribed and also get translated to form the enzyme permease. So gene Y, it will code for enzyme permease. As its name is suggesting us that permease, it is related with the permeability. It is related with the permeability. So this enzyme permease, it will increase the permeability of the cell to take more and more lactose. Okay. So what is the function of permease? It will increase the permeability of cell to take more and more lactose. Now come to the next structural gene, that is the A gene. A gene, it will again get transcribed, it will again get translated to form the enzyme transacetylase. This transacetylase enzyme, it helps in the transacetylation of lactose in its active form. Okay, so it will help in transacetylation process of lactose. Clear? So this is when there is absence of inducer, the lac operon model is switched off, and when there is presence of inducer, we call it as that it is switch on. I hope the lac operon model is clear to you. Clear? And this kind of regulation, it is called the negative regulation. I am again repeating this because it is controlled by the repressor. Okay, lac operon, it can show positive regulation also, but that positive regulation is not in your syllabus. In your syllabus there is only the negative regulation, and both these diagrams